giving a reminder, right?
Yeah, correct.
Okay. Presentation and 10 minutes.
So Ocean, I have lost that co-host access. If you want, you can make me the co-host if needed. Like,
Yeah, so I just actually took it because I had to stream,
yeah, right,
and
right.
it wasn't giving that streaming option.
Right. Welcome. No one, Zoom meeting.
I was actually on the host access that time because I shifted from main account to my secondary one. So you can give me the co-host.
This
I'll
is
just
the
keep
new
that.
task.
I've made you the co-host now. Okay, before we begin, just to quickly give everyone some instructions, please mute yourself when you are not talking, when someone else is presenting. The moment that we reach 100 participants in this group, we will be sending the YouTube link to the live of the Journal Club to all the emails, and whoever is not able to join here will be able to join there. I'm just mentioning that. And yeah, Sahaj, you can start in, actually you can start. We are 3 minutes into it. So yeah.
Just give me a second.
Yep.
Can you give me this for this?
You need co-host permission?
I need to share my screen for that.
Yeah, okay.
Okay.
Okay, everyone, I think we'll be starting in a bit. Just so I know, from where is everyone joining? India, Georgia, Bulgaria? please write that in the chat so we know from where people are joining, which time zone. Okay, we have a few: Georgia, Turkey, China, Russia, Afghanistan. Okay. Okay. Nice. So there are a lot of people from outside India, which is good to know. I think that Harsaj will be joining us in a bit. If he is unable to join, then I will just share my screen and I will start presenting the
I'm
introduction.
here, doctor.
Okay, please start.
I can't really present my screen. I don't know why, but
What is the issue?
it's disabled for me to present.
Okay, excellent. You are the co-host, so I don't know why. Once again, can you try again?
I think yes. Is
Okay.
my visible now?
Yes, we can see your
All
screen.
right. So let's start the event session. Hello, everyone. Welcome to the Global Journal Club from the GCMER. So let me explain you before we start what is GCMER. So GCMER, or Global Consortium of Medical Education and Research, established in 2020 as a leading research research network in India. We have international operations now with the SRCs, which is the Scientific Research Councils, in more than 11 countries, operating with the research-oriented students and even the doctors globally. We lectures, YouTube videos, mentoring sessions, journal clubs, networking sessions, CV building workshops, and a lot more. So that was just a quick introduction of GCMER. For more information, you can follow us on Instagram at gcmer.hq or simply by scanning the QR code given here. Okay, so I would like you guys to visit our website as well, which is www.gcmer.com, or again, you can just scan the QR code mentioned here to visit our website. This is a glimpse from our website just to show you guys. It's a newly designed website which has membership part, a learning corner, and GCMER team, and a globalization corner. So as you can see, we have articles and posters from our international SRC members uploaded in this website. Okay, so talking about why are we here today, which is the Global Journal Club. So let me explain you what is the Journal Club very quickly. It's a group of medical students and the doctors and reputed researchers from all over the world who come together, and we meet twice a month, that is first and third Saturday of the month, and present the latest researches that are published in top journals, and then critically evaluate and review the paper to find out the gaps and some interpretations from the paper from the original research. Now talking about the structure of the journal club sessions. So we have basically we have three classifications. First is the presenter, who is a member of GCMER, then audience, who can be non-members, as well as the members. It's open to all. And then we have the mentors, GCMER experts, the doctors, or other invited guests from all over the world. Let's talk about the process of the journal club session and how it's going to be happening. So first one is the presentation. The speaker will be presenting a paper in the form of a presentation of PPT. And then the speaker, himself or herself will evaluate and give the analysis of the paper, and then we have the mentor's review. The invited mentor will critically review the paper and find the gaps and give a better understanding of the paper. And then we have the audience point of view. So the attendees are free to raise their hands and put forward their opinions, or simply ask some questions which will be clarified in the meeting. Next up, we have about today's journal club. This is the first journal club session, and we are discussing cardiovascular diseases. We have a first presenter, Bala Murali, who is from Tbilisi State Medical University, year 3. Then we have Sneh. He is a presenter from India, Government Medical College, Surat. Then it's me. I am Harsahej Vilku. I am the president of GCMER Georgia, and I'll be moderating today's session. Then we have Dr. Osinbet, who is the global president of GCMER, and she will be mentoring our session. So let me take this opportunity to introduce to you guys our Discord community. So you can scan this QR code and join our Discord community of international students, doctors, and other researchers who are into research, where you'll be also receiving the updates about the latest happenings of GCMER. And that's all from my side. Let's begin the research. Let's begin the session. And if you want to become a part of the presenters, then you are free to contact us anytime and become a member and get other membership perks as well. Or you can just drop us a mail at research.gcmer@gmail.com. So yes, that's it. And I would like Bala to start with his presentation first.
Thank you, Harsaj, and thank you for this opportunity. Also, I'm not able to share my screen. It has been disabled. Oh, perfect. It is viewable, right?
Yes.
Perfect. So, good evening, guys. I will be presenting the paper, the first paper for Journal Club for GCMER today. Now, my name is Bala Murali Krishna. I'm a third-year student from Tbilisi State Medical University, which is in Tbilisi, Georgia. The paper that I'm going to be presenting is the association of habitual alcohol intake with risk of cardiovascular diseases. Now, this paper was a conjoined effort, but the lead author was Dr. Kyron J. Beidinger. And it was published in the March of 2022 in the German Network Open Journal. The content of today's Journal Club, we will be discussing about the background of the research, the methods that was utilized in the research, the results, conclusion, and my opinion on the research, that is critical analysis of the paper, and the scope, whether there's any scope for this particular research later on in the future. Now, the background. So the rationale behind the study is the fact that cardiovascular diseases can be precipitated by habitual alcohol intake. Now, it is a factual thing because it has been previously proven via research data. What these authors would try to do is that they wanted to associate certain limits of habitual intake of alcohol with cardiovascular diseases, because USADA, they have an approved measure of how much alcohol a person can intake per week. For men, it's supposed to be 14 or lesser drinks per week, while for women, it's 8 or lesser drinks per week. Now, given this association, they wanted to try and implement a method in order to see if it was effective or not. And that can be considered as one of the rationales behind the study. Now, usually the previous studies all demonstrated a J or a U-shaped curve. So people without any alcohol intake or lifelong abstainers also showed almost a similar amount of cardiovascular disease incidence, but intake of probably like a drink per week or maybe 2 drinks per week showed lower incidence of cardiovascular diseases. They were trying to explain why this was the case as well. Now, there are 2 things that kind of limits observational epidemiology. That is reverse causality and the confounding factors. The major issue with a study like this is the ethics and the logistics which goes behind into the observational epidemiology. But utilization of Mendelian randomization, which is a method for overcoming this confounding and reverse causality, help them to aggregate a lot of data in a short amount of time, with, or what do you say, bypassing the ethical morality as well. Now, utilizing Mendelian randomization alone can cause a linearity in the graphs that we obtain from the statistical data. But to overcome this, they also utilize something known as NLMR, or nonlinear Mendelian randomization. Now, the methodology. Primarily, it was a cohort study, but it was conducted in two different countries, one in UK, one in US. The one in UK was conducted by the UK Biobank, while the one in US was conducted by the Brigham Biobank. You have a primary and secondary, the primary being the UK Biobank. Now, the total participants in the study encompassed 371,463 participants. Now, in the secondary, that is in Brigham Biobank, 30,716 participants were assessed and genotyped. Initially, 500,000 participants, upwards of 500,000 participants appeared for the study from the period of 2006 and 2010. What we're going to discuss next, as the inclusion and the exclusion criteria, help the authors to reduce the number of participants depending on what they're looking for. The inclusion criteria mainly involve the fact that the participants were of European ancestry to rule out any skew of the data, as well as the fact that only individuals with complete genetic data, social data, and family data were allowed to take part in the study. Exclusion criteria, the major one is the missing of, genetic data from the genotyping, as well as degree of close relatedness. If a family, if for example, two siblings showed up for the study, only one was allowed to take part because the data can skew depending on how, close these participants are. And subjects with missing outcome data, because some can be already predisposed to cardiovascular diseases, and which is not due to habitual alcohol intake. So they were also excluded out from this study. Now, the study focused on 6 phenotypes of cardiovascular diseases and 10 continuous variables. The 6 phenotypes were hypertension, stroke, atrial fibrillation, stroke, and myocardial infarction, as well as coronary artery disease. The 10 continuous variables that were focused on were systolic blood pressure, diastolic blood pressure, LDL values, HDL values, your total cholesterol values, apolipoprotein A values, apolipoprotein B values, C-reactive protein values, and gamma-glutamyl transferase values. And obviously, a 2-sample Mendelian randomization study was conducted, utilizing a package. Utilizing a package in the software, our software, known as the 2-sample Mendelian randomization. MR-Egger and MR-Presso, these terms are usually the packages which is utilized in the R software in order to reduce any horizontal pleiotropy, especially MR-Presso gives you a value which reduces horizontal pleiotropy. So it sees if there are any other confounding factors for the genetic instrument that we chose. Now, talking about MR, we also need to talk about the genetic instruments that were chosen. There were 2 primary genetic instruments which were initially hypothesized that would work for the study, that is AUD-R and AUD-C. AUD-R was selected as the primary, genetic instrument, while AUD-CR was selected as the secondary genetic instrument. So it was more for like a confirmed analysis. In NLMR, that is nonlinear Mendelian randomization, 2 methods were utilized as fractional polynomial and piecewise linear method, which we will be discussing once we get to the results section. The results. What the authors found were the fact that J and U-shaped curves were still followed, even though they did account for confounding factors. And this also reduced the cardioprotective actions of modest alcohol intake. So according to USADA, we could take up to 14 drinks per day, but that was considered to be a modest amount of alcohol intake. But even that is kind of ruled out as per the study. AUD-R, which is the primary genetic instrument, is closely related with alcohol intake. So it kind of ruled out most of the confounders, which were kind of a problem for the whole study. Genetic evidence also supported associations between alcohol use and increased risk of hypertension and CAD. So, as I said earlier, AUDR helped in this hypothesis, and also, in fact, it became a reality because AUDR helped in identifying the causes which went into hypertension and coronary artery disease with linked of habitual alcohol intake. NLMR better approximated the association between alcohol intake and both hypertension and CAD with a p-value of less than 0.001 for both models. Both of them significant. The reason why I only mentioned two of them is because the other four, phenotypes, the cardiovascular disease phenotypes, stroke, myocardial infarction, atrial fibrillation, and heart failure, all four of them didn't have a significant value, only showing significance in hypertension and CAD. They also suggested nonlinear association between alcohol intake and continuous risk factors only for 3 continuous variables out of the 10, that is SBP, DBP, and LDL levels, with all of them with a lower p-value of 0.001. Over here, if you look over here, you can see the first graph is a sex stratified graph associating between alcohol and primary cardiovascular disease outcomes. Now, the reason why this graph is important, and I included it in the presentation, is because if you can look over there, in men section, with a 95% confidence interval, the p-value is 0.0010, which is not significant amongst light drinkers. And light drinkers were approximately 57,690 people out of the entirety of the study population. The value kept on decreasing until it reached a significant value until the abuse of drinkers, where the p-value was lower than 0.001. Same goes for coronary artery disease amongst men, where the p-value was initially high but kept on decreasing until abuse of drinkers. What's even more confounding is the fact that hypertension amongst women didn't show any significant p-values, which could lead to the fact that it wasn't a good genetic instrument which was chosen for the study because it did not take into account how women reacted to alcohol or the effects of alcohol in their body. In the nonlinear MR test, what we need to look at is the quadratic p-value and the Cochrane p-value. A lower quadratic p-value suggests that the particular disease follows a nonlinear regression. So for hypertension and CAD, both of them we have a nonlinear, following of regression. But for MI, stroke, and heart failure, as well as atrial fibrillation, all of them have higher p-values than the associated p-value. And so we cannot say that the test was completely successful. The next one is the Presso value, where we MR Presso sensitivity analysis for the 2-sample MR, where we check for pleiotropy, horizontal pleiotropy. And the global test values of this is given over here. What we need to look at is the global test p-value. For hypertension, the pleiotropy is very high, it's 0.285, but for coronary artery disease, it's 0.019. It is still high, but comparatively, it is better. Also, for coronary artery disease, a single outlier was detected. Again, goes to show how the genetic instrument which was chosen for the study was not appropriate. This is for the continuous traits, but that's not of much significance because all the p-test values, except for one, that is LDL cholesterol levels, everything else had a very high p-value for global test value, which goes to show that the rest of the factors were kind of insignificant in the statistical analysis. This is the U and J-shaped graph that you obtain when you do statistical analysis for alcohol consumption and prevalence of cardiovascular diseases. You see how abstainers, even lifelong abstainers or probably abstainers after consuming alcohol, they still have a moderate amount of cardiovascular disease risk incidence. But it reduces when you take 0 to 10 drinks per, let's say, per month or per week. And it keeps on increasing when your drink count increases all the way up to in between 0.4 and 0.45. This trend is then, attenuated with Cox destruction values. That is, the black one is the Cox values, while the lighter ones are the adjusted values. These adjusted values also show that even modest amount or light amount of alcohol intake kind of predispose you to hypertension in men. For women, the same trend is again followed. This is the nonlinear MR analysis of alcohol and total mortality. This is the piecewise analysis, while this is the binomial fractional analysis. The shaded region shows you the variance. This shows a high amount of variance due to a lot of confounding factors, and this is before adjusting. This is before adjustment. This is the polynomial nonlinear MR analysis after adjustment. It shows a much more lower variance, and it clearly shows that there is a trend which follows with alcohol intake and cardiovascular diseases. The conclusion which the authors came to was that light to moderate alcohol consumption may have cardioprotective advantages, but these effects are significantly masked by lifestyle variables. Now, this is very important due to the fact that they only attenuated for 10 variables. If a study is later on conducted, which assesses more variables, that would mean that even the protective advantages of light drinking might completely be ruled out. They were also able to, create an association between alcohol intake and cardiovascular diseases, but it was already created earlier on by previous studies. Now, my comments on this mainly is the fact that I'll talk about the merits of the study. Now, the merits, they talked about how they wanted to create a link between cardiovascular disease and habitual alcohol intake. They were successful in creating that link. Now, they were also successful in creating the link between modest and light alcohol intake to precipitation of cardiovascular diseases. This was not done earlier in previous studies, mainly due to the epidemiological studies, drawbacks. But utilization of Mendelian randomization, it helped them come to that conclusion. It also, as I mentioned previously, it also mentions that if you take into account a lot more lifestyle factors, it might mean that even light drinking can precipitate cardiovascular diseases. A higher amount of alcohol intake warrants higher risk of cardiovascular diseases. And the public health limit or recommendations of habitual alcohol needs to be reduced from 14 for men and 8 for women accordingly. Now, the demerits of the study, the main demerit is the fact that the genetic instruments that they utilize, the AUD and AUDCR, both of them are not for habitual alcohol intake. Instead, they're for acute or chronic alcohol intake, which is an extreme abusive amount, because AUD stands for alcohol use disease. Now, it's usually utilized for excessive amount of alcohol intake, but the study was focusing on habitual intake of maybe less or moderate amount of alcohol. Utilization of AUD and AUDCR kind of meant that the graphs were going to skew in a manner where it's going to show that the entirety of alcohol intake is going to be bad for a person's health. So it did not account for the fact that AUD and AUDCR does not account for a low amount of alcohol intake. And also this created pleiotropy, because AUD is also linked with congenital CVD, genetically predisposed CVD, and this pleiotropy was also not accounted for. And yes, that's pretty much it. Thank you.
Thank you, Bala, for such an informative presentation. Now, I would invite Dr. Ocean to kindly give your valuable feedback and share a critic analysis.
Yes, surely. Firstly, thank you so much. It was a very good presentation. Just one thing, yes, it would be great if you minimize the screen on the right side, because the right side was covered. Okay, I do have a few questions. Did you see any correlation between post-menopausal women and the CVD risk? Was this taken into account as one of the confounding factors?
Thank you, Dr. Ocean, for the question. Now, thing is, the postmenopausal women and CVD risk was never taken into consideration for the study, because for this study, they stuck to, let's say, BMI, your average weight, then your blood pressure, and your lifestyle factors, which involved your diet, because there was a question. So the initial part of the methodology involved the releasing of questionnaire to nearly 500,000 participants, and this spanned from 2006 until 2010. So this questionnaire contained materials which were relevant for dietary intake mainly. And I think that's one of the major reasons why the graph for women, especially when you do a sex stratified graph for women, is completely skewed to one side or the other. Because it either shows, especially the nonlinear graphs for women in hypertension, shows a graph where even people without even lifelong abstainers amongst women, they show a higher risk of cardiovascular diseases, much more than abusive drinkers. So I think the fact that they did not take into account a lot more confounding factors, the study was kind of skewed.
Yeah, I agree, because I would say that menopause and postmenopause and all the hormonal changes. See, I am not a cardiologist, but this is something that's basic knowledge, that these hormonal changes can affect your cardiovascular risk, right? Now, another question for me was actually, can you just go back maybe 1 or 2 slides? Yes, yeah, in the discussion itself. So my question is, why did the authors choose the AUDR questionnaire, the marker? Like, why did they choose that? Is there no other marker which is for like moderate drinking?
Unfortunately, given the limits of our scientific discovery, there is none right now. Because AUDR was actually chosen after significant consideration, because initially there were going to be 13 markers amongst AUDR, and amongst AUDCR, there were going to be 20 markers, 19 markers, sorry. Out of these 13 and 19 markers, it came all the way down to 4 and 9 markers, because they had to take out a lot of other markers which kind of represented pleiotropy, like more confounding genetic factors. So they stuck with the 4 markers of AUD and 9 markers of AUDC. And that's how they came to that.
Yeah, because as you mentioned, if you choose the wrong marker, then it
Absolutely.
automatically means that the results and hence the interpretation are going to be off, right? Because as we are making this recommendation that the required or like the recommended daily limit for alcohol intake for men and women, it should actually be reduced. But
Yes.
you are making this recommendation on the basis of something that was actually skewed. So again, this is just a lesson for all researchers that it's actually very, very important when we are choosing a study design, that we choose the right markers, we choose the right variables, and we also choose the right outcome that we are looking for, because that can change our interpretation. Thank you so much, Bala. I'm going to move to the other questions.
Doctor,
I think Anisha has raised her hand and Archana is going to ask a question in the chat. Yes, Anisha, go ahead.
Thank you, Vala, for the wonderful presentation. I loved it, the way you presented and your confidence. It was very good. My question was, so did you mention anywhere what was their limit of light, moderate, and heavy drinking? Like, what was the limit that they took?
I'm glad you asked that, because I couldn't mention this because my presenting time was quite short, and it's quite a lengthy thing to discuss, actually. So the light, moderate, heavy, and abusive drinking was from the limit of 0 to 8.4 was light, 8.4 to 15.4 was moderate, 15.4 to 20.4 was heavy, and 20.4 to 27.4, or even more than that, was considered to be abusive drinkers. Now, most of the participants in the study fell in the category of moderate to heavy drinkers, and a minority falling into abusive drinkers, and another falling into the light drinkers. And I just want to clarify one thing. When Dr. Ocean was speaking about choosing the right marker, she's absolutely right. But I believe the reason why AUD actually helped in the study for creating the link between alcohol intake and CV diseases, and also coming to the conclusion of reducing the limit of alcohol intake, is because even though it searches for abusive alcohol intake, it does have markers which kind of predispose you to normal, like moderate to high levels of alcohol intake. So the cardioprotective actions can be assessed using that particular biomarker. So I just wanted to say that. But thank you.
Just to follow up on that, so the limits that you mentioned was alcohol per week, like 8 drinks alcohol per week was light drinking. Am I right?
Yes, absolutely. And this limit, they came to the conclusion why utilizing, so one measure is 14 grams of alcohol. Now,
Okay.
the measure is utilized in US, while grams is utilized in Great Britain. So one measure is 14 grams of alcohol. So 8.4 measures is what they say. That is light
Okay.
drinking. So 0 to 8.4 measures, 8.4 to 15.4 measures.
So what is the value coming in grams? 8.4 measures is how much in grams? Did they mention?
They did not mention that, but I can give you the answer for that.
Like approximation, is it coming 14 gram only? Something like that?
No, it's not coming to 14 grams. 14, it would probably be around 117.6 grams.
Okay, yeah. Thank you for answering that.
No worries. Thank you.
If anyone has any questions or they just want to add anything to the discussion, you may go ahead raising your hand. All right. If there is no one who wants to ask a question, we shall move on to the next speaker for the day, who is Sneh. Sneh, you may
I think
share
there
your
is.
screen and start.
I think there's one question,
Yes.
Harshaj, in the chat.
Yes, yes. So, okay, so there's a question from Fatma. She's asking, she has a question. If a man above 40-year-old had previous cholesterol related problems, but he is still light drinker 2 times in a month, what are the chances of getting a cardiovascular disease? What's the risk factor, basically? Bala, would you please?
Thank you for the question. And it's actually a great question. Now, in the question, in what she asked is, the man is 40 years of age, which means age is also a factor in cardiovascular diseases amongst men. So the primary cardiovascular disease which plagues men is hypertension, right? So let's talk in terms of hypertension. And he already had a previous history of cholesterol. And he's a light drinker, not even a light drinker, he's an occasional drinker, which drinks twice a month or so. He would still be at a higher risk of contracting cardiovascular diseases, but it's not due to the drinking, because the major factor over here is the fact that he has cholesterol, and his drinking might only contribute to probably a percentage of why he might get the cardiovascular disease.
Thank you for the explanation, Bala. Any
I
other
think
questions, please?
towards the conclusion, what this study tells us is that to get an association between light drinking or casual drinking and CVD, there are enormous number of confounders. They try to reduce it, but still there are so many confounders and so many other things happening with CVD, that having a drinking like association between casual drinking and like a final thing that they will develop hypertension or CVD is really difficult. That's what I can conclude. Am I right, Bal?
As a matter of fact, you're absolutely right, because the amount of now I remember saying this in the beginning of the presentation. The epidemiological studies, like they're kind of confronted with two factors, that is the ethics and the morality. Now, the confounding factors are one of them is another factor which affects epidemiological studies, and the issue is the fact that these confounding factors cannot all be categorized. There are so many things that goes into an individual's life. So all of them can be a confounding factor which precipitates you for cardiovascular diseases. And due to this, they came to the conclusion, okay, utilizing a genotype as a proxy might help us study or reduce the number of confounding factors. But in fact, what they didn't take into account was utilizing a genetic instrument which already had a high pleiotropical variance for the study. So yes.
Thank you, Bala.
Thank you, Bala, and thank you everyone for the questions. If you have any questions, you can type in the chat box and we shall answer you for the same.
Thank you so much. Shall I stop sharing the screen?
All right, Bala, you can stop sharing your screen. Sneh, can you please start sharing your screen now?
Ocean, this is an exciting poll.
Is it visible?
No, can you share your presentation?
Yes. No. Now?
Yep, it's visible. Please put it on a slide share mode. I mean, slideshow.
Unfortunately, slideshow is not working. So,
All right, you can go ahead with this.
okay. So, hello everyone. Good evening. Myself, Sneha Sachani. I am first year government medical student at Government Medical College, Surat. And today's article is treating cardiovascular diseases with liver genome engineering. It was published at 1st March 2022 in Current Atherosclerosis Report. The authors are Aurea Hurley and William Lagor. Topics covered are method for gene editing, delivery vehicles, target for disruption or deletion, and recent clinical progresses. So moving on to background, rationale behind the study. So as liver plays a critical role in production and clearance of the circulating lipoprotein particles. Lipoprotein particles are major contribution to atherosclerosis, which is major CVDs. So we are examining the new gene editing approaches, which is specifically liver directed for treating the cardiovascular diseases. Methodology. So this paper is actually a literature review. It has 75 papers included in it. But the main problem with it is there's no clear study method description in the article. And so I've addressed it in my comments section. Moving on to results. So first topic is method for gene editing. The genes which are discussed in this gene editing topics are firstly the lipoprotein lipase, gene for lipoprotein lipase. Second is gene for apoprotein C2. And third is the gene for lipoprotein lipase activator. Coming on to methods for gene editing. The basic method for gene editing is clustered regulatory interspace short palindromic repeat or CRISPR associated 9 protein system. It uses a guide RNA for guiding the CRISPR to particular sequences where it creates a double. Yes. Yeah. So it uses CRISPR system. It uses a RNA guide RNA to guide the CRISPR system to particular sequences where it creates a double-stranded break. The major three methods, which are HDR, HITI, and prime editing. These three methods are based on the CRISPR system. HDR, it is a repair pathway that uses a donor template flanked by homologous sequences to insert new genetic materials. So, is it visible now?
No, your presentation isn't visible. Yep, now it's visible.
Okay. So HDR is a repair pathway that uses a template. Now, the basic principle behind this gene editing is creating a double-stranded break in the required gene, adding a new gene or replacing the unrequired gene, and so thereby creating a mutation, that is frame shift mutation, which leads to pre-termination of the gene, so that no unrequired proteins are translated. HITI, it is a method to replace or insert large sequences of DNA with CRISPR or Cas9. HITI is an efficient method than HDR, in which it uses CRISPR system to create two double-stranded breaks. It inserts a new sequences, which may be a gene or a nucleotide sequence or exon sequence, and thereby prevent the transcription and translation of the unrequired proteins. Prime editing. Short regions of sequences changed to any base using template-based reverse transcription for modified gRNA scaffold. So prime editing uses a sgRNA, which is scaffold gRNA, and thereby changes the base sequence in the
Hello.
unrequired.
Hello.
Yes. Hello.
I haven't.
Could you please mute yourself, uh, other than the speaker? No, you can continue.
Yes. So these are the three basic techniques for gene editing: HDR, HITI, homology independent target integration, and prime editing. In
Hello.
recent time, prime editing.
Hello.
Anyone's mic?
Sneh, you can continue. There's some problem with some participants' audio.
Okay, so the next topic is delivery vehicles. The delivery vehicles are the major deciding factor that a gene editing technique would fail or it will succeed. So the three major delivery vehicles are AAV, adeno-associated virus, lipid nanoparticles, and angiopoietin like 3. AAV, it is packaged with capsids, which are derived from the bacteria, almost all of which have high tropism for the liver. That is, capsids which have high integration factor into hepatocyte genes. So it can be used for inserting the CRISPR system, which can work to create the DSPs. Also, AAVs, the major advantage of AAVs is that it works for a long time, that is stable for months to years. So once the gene is included into hepatocyte, it will continue to work till months and prevent the accumulation of lipoproteins. Lipid nanoparticles. These are non-viral particles most often used for delivery of mRNA. Third, angiopoietin like 3 inhibits lipoprotein lipase responsible for clearance of triglycerides from circulation. Lipoprotein lipase is produced from hepatocytes, so it is required for the hydrolysis of endogenous triacylglycerides, which are major contributing factors to atherosclerosis. Next, recent clinical progress. So, in vivo application of CRISPR-Cas9 for human therapeutics is already in progress in a trial to treat Leber congenital amaurosis, deliver SA-Cas9 to retina. SA-Cas9 is Staphylococcus Cas9. So, it is the first trial which was somatic gene editing. In the category of somatic gene editing, it was the first trial which was done to treat the Leber congenital amaurosis. The first liver-directed gene editing trial was done to treat transthyretin amyloidosis. So Intellia, it initiated the first CRISPR or Cas9 liver-directed gene editing trial. Transthyretin is a protein that transports vitamin A or retinol form to the retina. So in the deficiency of transthyretin, that is mutation in the gene which produces transthyretin, it lead to deficiency characteristic of vitamin A deficiency in retina. So a liver directed gene editing trial was done. Mutation was removed from the gene and a new base sequence for the transthyretin was inserted using the HITI, and it lead to increase in the retinol transportation to retina and subsequently the effect on retina. Verve Therapeutics is developing a liver-directed therapy for FH, familial hypocholesterolemia, through disruption of PCSK9 using a base editor to avoid the undesirable on-target effects induced by DSP as well as LNPs to transiently deliver the base editor as mRNA. So recently, in the category of decreasing the lipoprotein. The first liver-directed therapy is VERV therapeutics. In this, there is a disruption of PCSK9, which is promoting the formation of lipoprotein lipase receptor. So its disruption lead to decreased intake of lipoprotein lipase and consequently reduction in the lipoprotein, circulating lipoprotein. This is the different method of gene editing. First is CRISPR-Cas9, gRNA, guide RNA, which binds to the target site. Cas9 produces a double-stranded break. HDR, it inserts the DNA template by creating the DSBs, leads to frame shift mutation. Third, HITI, it creates two DSBs and insert the donor template. And last one is prime editing, use of the sgRNA or scaffold guide RNA. This is the procedure of prime editing. So in the recent PCSK, that is VERV therapeutics, this was the studies, and the delivery vehicles used are AAV, LNPs, and lipid nanoparticles. HITI is more efficient than HDR in clonal populations. This was the estimation using the fluorescent. HITI showed more brighter fluorescent, and it was more efficient. That is CRISPR in vivo gene editing for transthyretin amyloidosis. First liver directed gene editing. This was the study, and this is the change in serum transthyretin concentrations in patients who received 0.3, that is the dose of CRISPR system. Major drawback to the system was the Cas9 Casper system used, which was derived from bacteria. So in humans, almost all of the humans have pre-existing humoral immunity. That is, it has both memory T cell dependent immunity and antibody dependent immunity. So this was the study done after taking the serum, showing the amount of antibodies produced against the Cas9. This was a human trial done in different races, showing the antibody present in serum. Conclusion. Many recent advances have been made in the field of gene editing for CVDs. Also, there are multiple attractive targets for liver-directed genome editing that could dramatically lower circulating lipid levels and reduce cardiovascular risk. Critical analysis. So HDR, which was a method. So the main disadvantage of this method is we have to use this method in the dividing cells. That is at the germline stage only. So it is impractical for adult liver because less than 1% of hepatocytes are actively dividing at a given time. So if we use it in the germline, inducing it in a single cell would not be beneficial. HITI generates a complicated mix of different editing events at the on-target site. So HITI uses Cas9 to produce two DSBs. So the Cas9, which uses guide RNA is not 100% efficient at identifying the target sites. So it may lead to production of DSBs at undesired site. And so if the template which is used in HITI, it is induced or incorporated into that undesired site, it would lead to mix of different editing events. Pre-existing immunity to SA-Cas9 and SP-Cas9. Risk of off-target cutting and insertional mutagenesis. This means the DSBs which are created by the Cas9. These are 100% dependent on the target site, but the deciding of target site, which is done by guide RNA, it is not 100% efficient, so it may lead to off-target cutting and insertional mutagenesis. My comments on this paper: there was no methodology section included into this article, so it was very unclear to read, and also the inclusion exclusion criteria was not included, because in the initial it was comparison, but later it has described the human trials, so the inclusion exclusion criteria for human trials were not included. Readability level, it was, as I'm first year student, so it took me almost 6 to 7 hours to study and understand this article, and then to create the presentation. So the delivery vehicles for gene editing, which I've described, it was actually at expert level, so it was very difficult to understand for undergraduates, and it was more directed towards the genomic specialists. That is, it has just the results of the 75 articles which are included in it, so no basic description of the any technique or any vehicle or the target genes was done, and so it was difficult to understand. Yeah. Okay, so thank you.
Thank you, Sneh, for a comprehensive presentation. So I would request Dr. Ocean to give a review on this.
Yes, thank you very much, Sneh. Firstly, very, very commendable effort. I know this is your first journal club, and it's quite difficult to summarize a whole paper which you haven't written. And it's also that you're in first year, and it's really very, very good that you presented this. It was most definitely an expert level paper. I wouldn't say just for undergraduates. I think that even for a doctor who is not in genetics or genomics, it would have been quite difficult to read. I do have a question for you. So, from all of this, can you very quickly summarize the top three recent most findings of gene editing that you understood from this? If you are saying something, then we can't hear you.
Yeah.
Yes.
Yeah. So it was not enabling, it was showing not. Okay.
Go
So
ahead. Yeah.
yeah. Thank you for the motivation, and sorry for this mistakes I have done in the presentation. I'm very nervous, still I'm very nervous. Sorry for that. So yeah, in this technique I've observed that the method HITI, so it can be used for long stable time. So if it is incorporated at the germline level, or at the embryo level, so it can reduce the chance of familial hypercholesterolemia, which is the most common causes for atherosclerosis. And second one, the prime editing technique. Uh,
Yeah.
it is also a very recent technique, and it has been used nowadays in most of the liver directed gene editing. It is used in it for editing. And lastly, the transthyretin amyloidosis, which was done in the human trial, it was successful. So
Mm.
it shows that it can be used for the cholesterolemia, and we can treat.
Understood. Thank you for summarizing that, because I think that will also allow everyone in the audience to understand what the paper was trying to say, basically. Okay, so now I'm going to let Anisha ask the question because she is, yes. Go ahead, Anisha.
Yeah, thank you, Sneh, for the wonderful presentation. I know how nervous you were, but I would like to give Siddhant a chance first to ask the question. Siddhant, you can please unmute and go ahead.
Hello, hello. I have a
You're
question.
audible.
I have a question. मेलोडी इतनी चॉकलेटी क्यों होती है?
So we already have people like this and they know that it's YouTube stream. Thank you, Siddhant, for your wonderful question. If you have any more, you can go ahead. But yeah. So thank you, Sneh, for the wonderful presentation. And what I would like to say is, I know how nervous you were, but you did a fabulous job in summarizing that beautiful article. What I just wanted to say was, so guys, anyone, do you know that from medical science, how can we go into genomics or like the laboratory research? If anyone has an idea or if anyone would like to know about it, you can just like type it in the chat if you know about it, that how can doctors go into genomics or lab research or write such research articles which Sneh has already shared today. uh, like you can comment it and that would be very good. I'll just say that so yeah, there is a laboratory research like after MBBS, we can the general thing that PG and super specialty, everything we can do. Another way, like Ocean can describe it better because she's a postdoc, and that is another way of doing science. Like we have, so I also got introduced to it a year back. We have almost familiarized ourselves with clinical research, but then there is another form of research which is laboratory research, and through which such articles come into being where they experiment in the lab and do a lot of experiments, and then they collaborate with doctors, scientists, and then we have all such articles out there. So that's another field, and if you are interested to know more about it, we can have a session on it, because some of the students do ping me sometimes that what is postdoc and how do we do that. So yeah, just wanted to give a heads up that this is how postdoc research happens, and this is what they do, like basically the lab research. Ocean, you can go ahead and comment if you like anything you want on it.
Yeah, always very happy to talk about research anytime, especially a career in research. So I have actually put up a poll for everyone because I know that gene editing has actually come into the light. Um, it has been in the light, I have to say, for a very long time. But very recently, it has come into the light because of its ethics, right? So people are questioning whether it's ethical, whether it's not ethical. But gene editing itself is not like you are changing the gene literally. So that's just something that is not known to a lot of lay people. And what I wanted to tell everyone who would like to come into the field of research is that just be curious. In your undergraduate time, there's really not much you can do beyond being curious, exploring all the research opportunities that you get, and trying to expose yourself to all kinds of research, right? Don't shy away from any kind of research. That is what the journal club is about. And that's all I wanted to say. Attend next time. I think, Harsahaj, we already have a topic for next time, right?
Yes, doctor. We have a topic for the next session, which is neurological disorders. So stay tuned for that. And yes, if you have any other questions, you guys can put up. There is a question by Sona, as I can see in the chat box. Where can we read this research article? Any website? If you mean this particular article, you can find this article in PubMed, or otherwise in general, you can read the articles in the search engines, which are, for example, Google Scholars, PubMed, and yep, so the PubMed is the most preferred one, preferably like my favorite also. So that's it. And how to get a certificate, which I'll be talking about. If you guys don't have any other questions, we can wrap up the session.
I think Bala has a question, and I will answer the question on how to
Yeah.
present here. So,
Okay.
people who want to present here, you need to be a GCMER member to present. You can, of course, attend as long as you do not ask baseless questions, in which case you will be banished forever from the Journal Club, because a lot of people here are spending a lot of time, putting in a lot of effort to actually, present their work, and it takes a lot to come into an audience of like 200 almost. So yeah, you can become a member. We will be circulating the post event form and the membership form for GCMER. You can surely join. Bala, you have a question, so I'm going to allow you to unmute. Yes, go ahead.
Thank thank you, Doctor. So my question kind of pertains to the previous article being discussed,
Yeah.
but I'm going to ask this question to Dr. Ocean directly. Now, like how my colleague Sneh has beautifully, presented about CRISPR, I wanted to ask you like the disadvantages and how we can address the disadvantage of CRISPR, which is mainly the pleiotropy of different variations of a particular gene being edited. So like the effects that we kind of don't account for, how do we kind of, keep track of all of those? And how can we get over that particular hump?
Yes. So firstly, just to address that, I am not a genomics expert, so I don't know how well I can answer this question. But what I can do is that I can tell you that there is a new technique which is in addition to CRISPR, actually. I do not remember its name, but I did read about it. I will most definitely get back to you on that, and I will send you the article on which I read about it. It basically does exactly this: is it eliminates the chances of pleiotropy, which occur very commonly with CRISPR and other techniques. And there are also other ways. So, for example, after the CRISPR has already been done, there is actually now AI which is trained to give you results which eliminate that. And it has been trained on many, many, many models of CRISPR. And that's also another way of doing that by sort of post processing the CRISPR data. This is usually very commonly used in things like EEG, in MRI, there's a lot of post processing. But now, even in genomics, this is something that's being done. And I will send you the name of that procedure as well. I will most definitely do that.
Thank you so much.
No problem.
Does anyone else have a question?
No, I think we are done with questions. And yeah.
All right. So thank you, Sneha, and thank you, Bala, for your amazing presentations, and thank you, Dr. Ocean, for sharing your valuable feedback. And so we have come to the end of the session, and let me take this opportunity to share with you guys the latest happenings in GCMER right now. So we have Equalized, which is a medical poster presentation. The topic and the theme for this is basically World AIDS Day, and we have the deadline for 20th of December. And so you guys can tune in and upload your paper, I mean the posters. And yep, and the second one is Alexa 2020, which is a national research conference which is going to happen in the GCMER Georgia, in the Tbilisi State Medical University. This is a abstract presentation conference based on the theme of cardiology, pulmonology, and gastroenterology. So we have abstracts and posters from all over Georgia, and they'll be presenting the abstract in the conference along with the expert panelists who will be assessing their papers and selecting the best abstract amongst them, and as well as the best poster. And the best abstract will be given the membership, I mean, the publication support from the GCMER. And just to mention, this event is in collaboration with the Tbilisi State Medical University of Georgia, and all the GCMER members are free, they have a free of cost entry. And in case if you are an international member, you will be receiving the email, I mean the links to the conference through your emails. and by your GCMER membership ID. So, and the third one is if you have any other questions or if you are interested to know more about GCMER, we have an induction session which is coming up on 20th again. So you guys can join us over there and get to know about GCMER even better. So I'll be sharing a post-event feedback form over here, and just let me just share in the chat box. So you guys can fill up this form and we'll be distributing the certificates according to this. And this form also has a link to the WhatsApp group, which you guys can join in order to, it's kind of like a community of the journal club, you can say. And the membership form is also provided by the GCMA official in the chat box. So that's pretty much it, I think.
Yeah, I'll take that question if someone want to understand more about the equalized poster theme. And also, guys, so please fill in the form ASAP because we'll be closing it by the end of today. So first of all, make a note of it that only the students who fill in the post event will get a certificate, because otherwise it's very difficult for us to track that who attended. And in the post event form, please do share your review that how do you find the journal club? Was it like, what was the best part and what could be improved going forward? Because this is a recurrent thing in GCMER. And obviously your review will help us in improving our journal clubs and making it more exciting. So please incorporate your feedback in the feedback form. That is what it is meant to be. Yeah, and you will get your certificates through the mail. So please keep checking on your mail. That is where we'll release your certificate. Explaining a little bit about the Equalize, the poster presentation that's going on currently. It is like a team event. And also we have opened it for individuals because we got some queries that we cannot get a teammate. So basically you can participate in a team from 1 to 4 now. And you have to find a research article on AIDS and just mention a conclusion of that paper. So this year's World AIDS Day was on 1st December, as you all know. That is when we launched our event. The theme was Equalize according to the theme of this year, World AIDS Day. What we are looking forward with this event is to understand the inequalities in AIDS. So you can find a research paper, which could be anything, and you can also take some example from our Instagram page. We have a sample poster there, and we also have some entries where students have put a lot of effort. So you can have a look at it. Basically, find a research paper, find a conclusion to that paper, that what inequality you feel is the most troubling currently in AIDS. Make a digital poster out of it and send it through our form. The form can be found on GCMER HQ Instagram page. And you can also like send a query to research or anywhere, and we can share that form with you. So you can send your entries directly there, and it is open till 20th. We have exciting rewards for the winners as always. And also do follow our GCMER Equalize page. So there's an Instagram page out there, and you can find it and you can see the entries, which are like superb. We have 4 entries and all of them are so good. They have actually put a very good effort of combining research plus art. That was the motive of Equalize, that you can put your creativity in a digital poster while putting a conclusion that's like a research in it as well. So go ahead and participate and DM us in case of more doubts. Thank you.
Thank you everyone for joining and attending this session. I hope you guys had a great session and it was very informative. So that's the end of the session. Thank you everyone.
Thank you.
Save the post-event form with you if you are not able to fill it currently, because as I said, we won't be able to track who participated and we don't want confusions that we participated but did not get a certificate. So yeah, please do that. And the form will close at 12. So just a reminder. Yeah, so as I said, the form is not visible to some people. Thank you everyone for joining. We'll just stay here for 5 more minutes in case anyone has any other query.
Yep.